There we go. Hi, everybody. Give people a couple of minutes to roll in. Hello, Valjean.
It's actually pronounced Valjean.
Sorry, say again?
It's pronounced Valjean.
Oh, how do you say again? Sorry.
Sorry, I don't know if you can hear me. It's Valjean.
Valjean. Oh, cool. Like French. Yeah, very neat. Sorry about that. All right. Double check where we're at. I think we're in the middle of a monohybrid cross based on my pictures. Oh, Daphne, that's a very cute picture of a cat. There's a cat. Lots of cats. Maybe. And massive cats. All right, get everything in place. Okey smoky, might as well get going. So we need to finish off our monohybrid cross. Not with that pen, because that is running out. All right. So actually, I can't remember when. I think Wednesday lab has already happened. I do believe. I'm not sure which of you are in the Wednesday lab session. Oh no, that's in the evening. Is it? No, that's Tuesday. So I think all of, hey, go on, sorry.
Wednesday lab starts at 6 p.m.
Oh, does it? Poor Gabby. She's got the short end of the stick this semester. And, you know, whoever is taking at that time too. Okay, so let me get chat up as well. I can see that at the same time. Oh, I'd love to see better pictures of your cats. On my screen, your cat pic is like that big. So it's kind of a little bit hard to figure out what is cat and what isn't cat, to be honest. You have to send me some pictures. Oh, and also, Richard, your pup, forgot his name off the top of my head, looks so similar to my Pepper, just broader in the face. So that's really interesting because I'm always trying to figure out what exactly Pepper is, because she's a bit of everything, I think. All right, so we left off sad cat. There you go. With our F1 generation. So hopefully this should benefit anyone who's taking lab tonight. So F1, actually, let me double check. What letters did I use? I think I used B's. Yeah, I did. There we go. All right, so here we go. Question for the class. What is the genotype of the F1 generation? Always. Without fail.
Heterozygous.
Correct, thank you. Valjean? Hey, there you go. Thank you, Richard. You too. So, heterozygous, we have a single dominant allele. Therefore, the phenotype is always that of the dominant parent, right? So this is a black cat, even though it isn't entirely black. So one thing that I like to teach my students, and I start with the monohybrid because it's easier, is always write down what gametes you can get from an individual. So in a monohybrid, it's a little bit redundant. It's like super easy. When we get to the dihybrid, it will help you a lot and it will keep you on track so you don't kind of trip yourselves up. So there are two different options now with the P0s. It's either dominant or recessive, right? From one parent. You can't get different. So dominant from the dominant, recessive from the recessive. Heterozygotes, now we have two different alleles. So we have two different potential gametes, one with a dominant allele and one with the recessive allele. All right, anybody remember what the law of segregation states? One of
The
Mendel's
alleles
three.
will segregate randomly.
Right, thank you, Valjean. So which allele each gamete, right, obviously produce loads of gametes, but every time a gamete is produced, which allele any given gamete has is random for a gene. That's the law of segregation. So if this is a random process, what do you think the ratio of these two different possibilities are? How many of this are you going to get to how many of this? If it's random.
Would be 1-to-1.
Yeah, exactly. It'd be 1-to-1. Yeah, it's like flipping a coin. You've got a 50-50 chance of getting one versus the other. So, and this will come important later in dihybrid cross and also in lab when we talk about our back cross. So Richard, what you're probably thinking of is way later in the F2 generation in terms of phenotype ratio, but we haven't got there yet. We're talking just about the gametes that contribute to the F2. We haven't actually gone through fertilization yet. All right, so I'm so looking forward to getting my lovely big whiteboards back. This is like equivalent of teaching on a postage stamp, which would be a challenge, I guess, not necessarily one that I'll be super down with, but there you go. Professor Olympics or something, I guess. So what happens next is those F1s get crossed with each other, right? I'm just going to draw it small because it takes up less space, and space is a premium. So we have heterozygote. Anthony, yes, that is 100% correct. So at the beginning, so this is where we're kind of picking up from, beginning of every monohybrid or dihybrid cross, the parents are always homozygous. Homozygous recessive or homozygous dominant, one or the other, right? Good stuff. So we also have over here, and I'm going to draw this in a square format because, you know, Punnett Square and all that. It's actually named after a guy called Punnett, believe it or not. Imagine how they taught genetics before. I think his name was Roger Punnett. Can't remember. But anyway, what would they call it before Punnett Square? I don't know. Mystery. So write down the gametes. Like so. Same for each F1 because they're all identical. And so far, so simple. And then fertilization is simply add these together. That's it. That is literally what fertilization is. And these often come out as betas, unfortunately, when I'm drawing on the whiteboard. So you add these two together, gives you homozygous dominant. So over here we have one of those. Going to write out the genotype ratio first, then when I've done all of them, going to write out the phenotype ratio, which is what we really care about. Add these two together, heterozygote. These two together, heterozygote. Technically, if we wanted to care about which chromosome, which allele came from, this would actually be the other way around. It'd be little b, big B. But we don't care about that too much because we just care about the genotype at the moment. We don't care about which parent, which allele came from. And then the final one over here is homozygous recessive. So we have one homozygous dominant, two heterozygotes. Ooh, if I could just reach. And one homozygous recessive. But we, in a monohybrid cross, in Mendelian genetics, don't care about that. We might care about that in some other weird form of genetics, such as codominance or incomplete dominance, but we're not talking about that now. We're talking about Mendelian genetics, right? And so now what we do is we look for the dominant allele. Wherever you have a dominant allele, you have a dominant phenotype. My youngest daughter was just telling me this morning about how this character in The Walking Dead has a tiger and then fashioned his dreadlocks into the shape of cat's ears. Now, if that were me in an apocalypse and I saw something like that, I'd probably just laugh. Not for very long, because then I'd get eaten by a tiger. But first thought would be to laugh, because that sounds silly. All right, and then the very last one would be homozygous recessive. There's no dominant allele to mask the recessive alleles. You'll see the recessive phenotype expressed. Ooh, we did this as quasi red, right? So these are, I only actually had a purple. Okay. So that is purple. Purple and black are actually kind of awful colors to use together because they look really similar. So you just have to bleed them. All right, so that gives us, if we add those up, one recessive, three dominant. And this is the ratio that you should always get in a F2 generation, right? So these are the F2, pardon me, the F2 generation of a monohybrid cross. Always and evermore. And this is also, given that you expect this ratio, this is what you'd be using if you were to, say, do a corn genetics lab, count kernels in a monohybrid cross, and then do a chi-squared test. This ratio, or if we expressed it as a fraction, right, which is that, you can see that down there, that would be your expected fraction of the total. You'd expect 3/4 black cats, 1/4 purple cats. Say you had a whole load of cats. And you'd need a lot. It'd be a very tired mama cat. All right, so that's a monohybrid cross. It's relatively straightforward. You only have one gene and two alleles. But the reason why it's so important to get this is because a dihybrid cross is almost exactly the same. The only difference being we have two genes. So the principle's the same, the process is the same, how you do it is the same. So if you know how to do it with a monohybrid cross, bless you, it's really straightforward to do it for dihybrid cross. Just follow the steps. right? Tortoise, one of the two, versus hare kind of deal. Don't jump ahead, follow all the steps, can't get it wrong. So now we're going to up our game a bit. Bing. And we're going to do a dihybrid cross. And anyone has any questions, as always, just throw them up in the chat or unmute and ask them. I don't mind. I like being interrupted. Okay. So now we have two traits, right, which are going to be controlled by two genes. Each of those are going to have two alleles and two phenotypes. So I'm going to choose fur color again, because we're already familiar with that. I've got a pen. So fur color and fur length. So these are going to be our two traits. I don't know what you'd be. B and L, let's say. And actually, this is, so fur color is at least in part controlled by what allele you have for melanin production. So you either produce a really dark melanin, or you don't, and you see an underlying lighter reddish, kind of reddish orange melanin color, reddish orange brown kind of sort of thing. Fur length is actually controlled by what is it? Fibroblast growth factor signaling, so FGF signaling. And essentially what happens is that doesn't get turned off, right? And so it keeps telling fur to grow longer and longer and longer, so you get long fur. Normal fur, wild type fur, is short. I don't know if that's why you see more short-haired cats around. I don't know, actually. Certainly in Texas, right? You wouldn't want to be a long-haired cat in Texas. That would be horrible, right? So we have two different traits, each controlled by a different gene. And we have two alleles for each of these. Man, I didn't leave myself enough space there. There we go. So we have, as before, black, which is the dominant, and red, purple, because that's the only pen I've got, which is the recessive trait encoded by the recessive allele. For length, we have short, which is the dominant, and long, which is the recessive. Now, if I didn't tell you this, right, I didn't tell you which of these phenotypes was dominant and recessive, how could you know? What would you look at to look at their phenotypes to figure out which of these two phenotypes for each gene is the dominant one?
Will you look at the F1 generation?
That's exactly what you'd look at, right? Because the F1s are always heterozygous for both, or any, however many genes you're looking at, heterozygous for all of them. Single dominant allele equals express the dominant trait. Whatever phenotype F1 has, that's the dominant trait, right? So I could, and probably will, ask you a question like that in the exam. So we have 4 different phenotypes, essentially. And so what you're going to see in an F2 of a dihybrid cross is not two different phenotype combinations, dominant and recessive, but four. All right, so this is where stuff gets a little bit more complicated. But as long as you follow it through, it's no harder than a monohybrid cross. And I get to draw furry cats, which is super fun. And again, I'm going to put both dominants together. Oh, sorry, Richard, just saw that. So there are four different phenotype combinations in the F2. There are four different alleles, you're right, but each individual, it kind of complicates things thinking about it that way because it doesn't really work like that. And you'll see why in a moment. All right, so we have P0 as always. We have our that one hasn't been in fine. Just we have our call it wild type, I guess. Normal, dominant, whatever works for you. Cat, which is black, and that's a terrible bit of writing. There you go. And short fur. Right. So this one, as always, is going to be homozygous dominant for both. And it's going to have that genotype. So again, this genotype equals this phenotype. Right, always goes in that direction, even though sometimes I write it the other way around. Biologically speaking, genotype, then phenotype. And this individual is going to be crossed with, this is where the fun starts. Though sadly, because of space issues, I can't draw full shaped cats all the time, which is rather sad. All right, so this cat is purple. Should be red, but it's purple. And it, which is a recessive, the recessive phenotype for fur color, and it's also long hair. And then you got super furry paws, fur everywhere. There you go. That is a long-haired purple cat. So it is, where would I put that? Put that over. Purple and long fur. So it has the opposite to the other cat. It is homozygous recessive for both, and it has that genotype. All right, have a think while I'm having a drink. What different, well, what are the gametes going to be from these individuals? What gametes is the dominant parent going to produce? Go on, anybody can answer.
Big B and big L.
Right, big B and big L. Yeah. And here lies one of the pitfalls or traps that students sometimes fall into. Gametes are haploid. They only have one allele of each. Frequently, I'm not entirely sure why, it's like some kind of cognitive dissonance, I guess. Students will actually give gametes two of the same letter. So you'd have a big B or a big L. So rather than the split occurring here, right, in terms of meiosis, which would give you essentially two gametes with big B and big L, instead, students quite often put the split there instead. So they're separating out the genes, not the alleles, right? Don't fall into that trap, because that will totally mess everything up. You're going to have always one allele for each gene. So if we had a tri-hybrid cross, you'd have 3 different letters. You'd have one of each letter, right? Dihybrid, 2 different letters, one of each. And so I don't see this as common as I used to, because I, make a lot of effort to head this one off, but I do see it occasionally, right? So try and keep this in mind and don't fall into that trap, right? Because nothing will work afterwards if you do. All right, so that's the first generation, the P0 of a dihybrid cross. Oh, I forgot to put up a what to do slide today. Never mind. It's a pretty straightforward day. All right, everybody good so far? So far, so straightforward. I'll be so tempting just to leave that out there. Never mind. Okay, so continuing on from the previous slide, we have these gametes, and you can never get anything different from the parents because they're homozygous. That's the whole point of true breeding P0s. You can only ever get one type of gamete from them. And so these two combine into the F1, and that gives us big B, big L, little l. Right? And even if we didn't know that the F1s are always expressing the dominant phenotypes, we'd be able to read off the genotype and see dominant allele for each dominant phenotype or phenotypes. That's a somewhat stern looking cat. I'm just going to draw the paws blank. I'm being a little lazy. All right. Now things get trickier. Now I want you all, and all of you, I'm assuming you're all there, but I want you to think, what are the possible gametes we can get from these F1 individuals? All right, so have a think, write down what you think. I expect more than just one of you, although I appreciate it, Valjean, sorry, to answer, right? You got to be engaged in this because you will be doing these in the exam, right? And there'll be a lot of questions about mono and dihybrid crosses and the statistics that go with it. Wow, that was quick, Israel. And Adriana, I should have like virtual candy or something to give out. Okay, so both of you are right. And what both of you have done is also the order that I would do it in. Start with the first gene, B, in this case, and start with the first allele in the genotype, right, which is the dominant one. So we have 2 different alleles for 2 genes. 2 by 2 is 4. right? Remember we talked about 4 different phenotype combinations? We've got 4 different allele combinations in the F1. And so for the dominant, you get, hopefully you can see that down there, 2 possible combinations for the dominant allele. You could either have, for the other gene, a big L or a little l, like so. Now you do the same for the little b. So little b can either have the same big L or little l, like so. All right. So now we have 2 genes. Can anybody remember what the law of independent assortment tells us? Doesn't apply to a monohybrid cross, only applies to a dihybrid cross, or more than that. What is the law of independent assortment all about? So if we're talking about a dihybrid cross, how many genes do we have in a dihybrid cross? 2, right? What does the law of independent assortment state about those 2 genes? Oh, wow, that popped up somewhere else. Thank you, Brianna. That's right. It's weird how that went up there. Super strange. Yes, Brianna and Richard. One gene has no effect on the other. So which allele you get for one gene, B in this case, has no effect on which allele you get for the other gene, L in this case. All right. What about the law of segregation? Another bit of recap. What does that tell us about the alleles for any given gene? Let's say B in this case. Do your ovaries or testicles choose which allele goes into which? No, thank you, Sarita. They segregate randomly, right? Keep in mind the big picture of sex, fertilization, sexual reproduction is random, right? So which gamete gets either big B or little b, totally random. And for whichever allele you get of one of these, big B in this case, the other gene, which allele you get, is also random. These 2 genes are independent from each other. So take both of those things all together and think, what is the ratio of, if you were to take, say, 100 of these gametes from this black cat, how many of each of these possible gametes would you expect to find? Either numbers or a ratio, doesn't matter which. Well, okay, so let's think about this differently, Richard. If you had 16 gametes, how many of each of these would you expect to find? Going to be 4, right? Or as Anthony said, if we have 100, a quarter of them will be this one. A quarter of them will be this one, 25 out of 100. 25 out of 100, 25 out of 100, right? So if you want to boil that down to a simpler thing, you have a 1 to 1 to 1 to 1 ratio of these possible gametes. Now, again, whenever you produce a gamete, you only get, well, I mean, technically you get 4 gametes, which kind of confuses things a little bit. But for each given gamete, it is going to be one of these 4 possibilities, right? So as an individual gamete, you can only have 1 genotype, right? You can't have 4. But whichever one you get, the probability of getting that is 1/4. Right? So this is kind of leading into the whole probability stuff that we're going to talk about once we've got through this. I think that'll probably be Friday. I'll have to double check the schedule. And this ratio is also something that you're going to see again in lab, because depending on how you do the cross, if it isn't a dihybrid cross, but say a back cross, which is what you're going to be doing with the flies, this ratio becomes super, super important. All right, everybody with me so far? Okay. So this is why I like going through this slowly, because if I just went ba-jam and put everything up on the board, you'd go, what the heck is going on, right? But this is so straightforward. Just follow all of this and you'll not get it wrong. Okay, now it starts getting a little bit wild. So as with a monohybrid cross, we have our F1 up here, right? Genotype is, let's see if I can move this a little closer. Genotype is essentially the same as what we did before, like so. Phenotype is what I've just drawn there. Sorry, my Alexa's just started playing a music song for some reason. Alexa, stop. I've never had this issue before teaching. I don't know what I'm saying, like if Mendelian genetics has like some kind of funky trigger function for Alexas. But anyway, so let's write out the alleles again, and you'll see why in a little bit. And I like writing these things out because you get to see what you're doing, right? Sometimes I do make mistakes, but as long as you see like what you have up in front of you, those mistakes become really obvious and easy to correct. All right, so those are, again, each F1 exactly the same. Oops. And you can do this too. In fact, I'd strongly recommend that you do so. But what I want you to do, to begin with at least, is I want you to join these together. Write down all of the genotypes for all of these 16, 4 times 4, 16 possibilities, right? I'm going to do it, but you should be doing it too at the same time. So this one, big B, big B, big L, big L. Oh, hey. Forgotten a allele there. I didn't mean to give you a demonstration of how you can see your mistakes quite so soon. I'm going to blame the acute angle of my whiteboard. That'll do. You know, it's funny, I've done this so many times, I should be able to just recall this from memory by now. Ah. And it's going to be a stretch doing it all the way over there. But let's see. And this one's easy. All right. So I'll give you a few moments to complete that yourselves. And I don't think it would be a bad idea in terms of studying to just make up your own. If you have cats, do cats. If you want to do aliens, do aliens. It doesn't matter. You can just make up whatever you want. It's a thought exercise at the end of the day. And see if you can repeat this. Right? You know, you could do, I don't know, green versus gray aliens with either like eyes in their head or eyes on stalks, for example. As long as you're consistent about what you choose as being dominant or recessive, you should get exactly the same result. I don't know what my kids are shouting about upstairs. So now we get to do the phenotypes, right? So these genotypes, we don't ever really worry about these so much because there's 16 different combinations, homozygous, heterozygous, all over the place, right? What we care about is what you can see, right? And so I don't know what's going on up there. It's either a cat puking or a dog eating some cat puke or dogs doing something that they shouldn't be doing. A funny thing to say, if you ever driving through the area where I live and a Wi-Fi network pops up on your device called the Animal Madhouse, that's us. Now I can hear one of my other cats busy digging away at one of the doors. It is never a dull moment, that's for sure. So all I'm doing is I'm going through here, and the pattern's always the same, which is why I can get away with this a little bit. But all you're doing is you're looking for a dominant allele. If you have the dominant allele, you have the dominant phenotype for that gene trait. There's some more around here somewhere. That one too. It's a lot quicker drawing cat heads than it is full cats. A lot more consistent as well. All right, so these are the same phenotype combination as the dominant parent. So we have black fur, short hair. And if you count them up, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9. Hang on. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. What have I done here? Oh, hee. Done something wrong. Yeah, thank you, Israel. See, you'll always be able to figure out what mistakes you're making because they'll be right there in front of you. So I think that's probably the first time I've ever done that. Anyway, that gets you 9, right? So 9 of these individuals should, so that was me being complacent, thinking I could remember the pattern, should be expressing the double dominant parental phenotype. Now, at the other extreme, right, if we're just looking at, and I do this different every time, but we're just looking at the other, question for the class, which genotype on here will get you the other parental phenotype combination? That'd be a nice quick question for the exam as well. I have to remember that one. Yay! Thank you, you two, Anthony and Richard. It's this one down here. That one. So that is purple. If you do, I don't know why I bother drawing the ears on. And long fur. That will get you 1. So of the total, and I think this is almost always a question I ask, one form or another. Of the total number of possibilities, give me a second, Anthony, I'm getting there. Of the total number of possibilities, 10 out of 16 or 5/8 of them are going to look like the original parents. Now, getting to Anthony's very pertinent question. What about the others? All right, so now we look at these. Just put R. Now we have 2 new combinations of phenotypes. We have black cats because they have a dominant black allele, but they're homozygous recessive for fur length. So these are going to be black long fur cats. They look a little bit like the coal puffs out of Spirited Away in this, but that's fine. Don't know if any of you have ever watched that film. It's really, really cool. And then we just go through and we find the other ones. So here's another one. And here's the third one. Like so. So that gets us, oops, wrong place. Alexa, stop. 3. And we get the opposite, right? Because, you know, you have to have the opposite essentially of that, which is going to be purple, because they are, this one is, if you can see what I'm, oh yeah, there it is. Homozygous recessive. Oh, hang on, Anthony. This one here is homozygous recessive for the fur length gene. So you see the recessive phenotype for fur length, but it has a dominant allele for fur color. So it's going to have essentially one phenotype from each parent. So it has the black fur from the dominant parent, but the long fur from the recessive parent. So this is a new combination that you don't see in the parents or the F1. Did that help? Cool. And then the final one, we said over here, now we are homozygous recessive for the fur color, but we have a dominant allele for the fur length. So these are going to be recessive for color, dominant for fur length, which is going to be purple and short, like so. And that gets us, I have to draw a little small, and we'll get 3 of those. So that's what gets us the 9:3 to 3:1 ratio. And these are new combos, new combinations, right? Ones that you haven't seen previously. Yes, that's right, Richard. So that's why when you go through this chart, yeah, I knew what you meant. When you go through this chart, you have to keep your eyes open for dominant alleles for both of the genes. If it's dominant for one gene but not the other, you're going to get the dominant phenotype. Which one? Like this one over here, for example. I think that's the genotype that you wrote out. This is has a dominant allele. You only need one. A dominant allele for fur color, but it's homozygous recessive for fur length. So it'll be black, that's dominant. Long fur, that's recessive. Black long fur. That's where I tripped up over here and made my mistake because I wasn't actually reading off of the genotype. So again, if you do all this, it's slow. But if you do it like this, you can never get it wrong. That's as close as I can get to a promise in biology or teaching in general. All right, I'll take pictures of this and get those up after class. And I am almost positive. I have to check to make sure. Oh, I think we might need to briefly talk about a test cross as well. That's kind of popping into my head, which is a different type of cross, but it's something that we can use to figure out genotypes. But once we do that, then we'll be getting on to the math part of this. And that's really all about what you expect from any given combination of genotypes. Right? Okay. Thanks very much, everybody. That was a lot of fun. See you on Friday, and stay warm and safe.
Professor,
Hello.
do you have office hours by any chance?
Not formally, but if you want to hang
No.
on, I'm happy to stop the recording and we can just keep on chatting.
Gotcha.
You got time now?
Yes, sir.
Okay, I've got like 40 minutes before a friend comes around to have a chat. Let me just stop this.